So how does it actually work? So basically the primary hypothesized mechanism of action is as an electron donor between complex 1 and cytochrome C of the electron transport chain. If you guys remember from biology, the electron transport chain is what goes on in mitochondria and enables you to produce most of your ATP. So when you have these kind of proton donors, I think you might remember the acronyms NADH and FADH2. They donate protons to this kind of like intermembrane space that kind of like proton buildup is what actually like drives ATP production and ATP synthase. It's basically a battery. You create kind of like a concentration gradient. So what is supposed to happen is electrons are supposed to flow between one complex and the next. The problem is some electrons leak. So the proton gets passed and the electrons, rather than going through all of the complexes, they actually leak into like the mitochondrial matrix. And they combine with things like oxygen and they produce things like superoxide and hydrogen peroxide, which you may have heard of before. Why is that a bad thing? Because those are very reactive oxygen species. Oxygen is a super reactive element, and it basically goes and kind of wreaks havoc in your body, specifically at the like protein level. So we'll go and oxidize a ton of proteins, denature them, and there are a lot of theories around aging that basically come from reactive oxygen species as the basis. So basically, in layman's terms, people think that ROS is like a huge contributor to the aging phenotype. And so the thinking with methylene blue is it can be ionized quite well. It accepts these electrons from complex 1 if they get leaked, right? If they don't go into the inner membrane, it accepts the electrons and passes them to cytochrome C. So basically, it directly kind of like shuttles them from one to the other. This is the hypothesized mechanism. And there is some solid evidence that this is the case.